From Microbe TV, this is Immune, episode number 11, recorded on August 15th, 2018. I'm Vincent Racaniello, and you're listening to the podcast about the body's defenders against disease. Joining me today from Ithaca, New York, Cindy Leifer.
Hi, and welcome back.
Summertime is winding down.
Oh, it's still nice here, although the summer has been extremely rainy this year, and we have hardly had time to enjoy it.
It has been rainy here, too. Yep. You're not too far from me.
Yeah, we had a lot of floods in the central New York area over the last 2 days. It's been crazy.
When are your undergrads coming?
They're already arriving. They are coming. They are. The main move-in weekend is this weekend, and classes start next week, Thursday.
Yeah. It goes quickly, doesn't it?
Yeah. How about where you are? When do they start?
The med students are already there. I went to medical school, of course. And they had their white coat ceremony a couple days ago, right? Where they all give them a white coat. They take the Hippocratic Oath and all that stuff.
Yep.
And the undergrads will probably be coming in a week or so. Yep.
Yeah, our veterinary students arrive about a week or two earlier as well. They started this week classes.
Today's just me and you, Cindy.
It is, sadly.
Sadly, the needs of being a grad student have taken their toll on Steph, so she will have to step out today. Hopefully she can come back next month.
Hope so. Yeah. Writing the thesis is not an easy task, so.
No. And we can't ask her when she'll be done, right? That's the question you don't ask. All right, good luck with that. But we do have a paper for you.
We do. Which
Cindy's going to tell us all about.
I will. So today we're going to talk about some really cool things. Immunomimetic designer cells protect mice from MRSA infection. And don't worry, I'll decode most of what that says. This is an article that was published in Cell in July of this year. And the lead author is Ying Liu. And the last author, why don't you take it? Because it's a Swedish name, I believe, and I don't know how to say that. Martin
Fusenegger.
So they're in Basel, and also in France and China are somewhere where the other group is located. And I picked this paper because I think it's really an interesting approach to a major problem. And so if you listen no further, the main point of the paper is that they use some cloning techniques and some engineering approaches to make a system, which they call a closed loop system, where they encapsulate and implant cells that they have designed to specifically detect microbial infection. And in this case, it's MRSA, and I'll explain what that is. And they are designed to produce a highly active antimicrobial molecule specifically targeted against the class of bacteria that MRSA is in, and that lyses the bacteria. And so the idea here is that this can be used with implants. So different implants are joints, valves, replacement devices in the body to prevent and eliminate infections. And
in a way, it's not so much different from CAR T-cell therapy. You're engineering cells and putting them back, right?
Exactly. You're engineering them to do a specific thing. And so the really cool thing about this is the authors took what they knew about immune responses and they engineered cells to specifically do what they want them to do when they want them to do it. And it's cool because when we put implants in the body, clearly they're not originally supposed to be there. And sometimes you can have hijackers or what do you call it when they're on the ship with you? Stowaways, I guess. Yeah, stowaways on an implant where the bacteria are associated with that. And you need very few of them to cause an infection. And they attach to the implant and they form biofilms. And this makes them resistant to antibiotics. And so often the only way you can cure these things is surgically remove them and put something else back in, which when you're opening a person up leads you also to potential infection again. So in this particular case, when we're talking about implants and introducing bacteria that are resident on the skin, it's a problem. But antimicrobial resistance in general is a huge problem. And it's an NIH high priority area. And the idea here is we've been using antibiotics to treat bacterial infections, and it's been extraordinarily successful for the last 100 years or so. But the bacteria are now developing resistance. And we're in big trouble if they develop resistance to all of the currently available antibacterials, then what do we use? And so this approach is completely different, doesn't depend on drug treatment, and it could be a really interesting way to approach such a problem. So the cast of characters in this particular paper include Staphylococcus aureus, or Staph aureus. This is a really frequent pathogen, and it causes lots of ER visits and hospitalizations, causes boils and pimples and all kinds of other things on the skin. And S. aureus is what's called a gram-positive coccus. And so the people who listen to TWIM probably have heard about this before. Gram staining, for those of you who aren't familiar, is a diagnostic stain, and it's based on the composition of the bacterial cell wall. So the bacteria has its cytoplasm, and then it has its cell wall, and then if it's gram-positive or gram-negative, might have a thick cell wall or a thinner cell wall. And when you try to stain these things, the stain goes in, and if they have a thin cell wall, the stain comes back out, and if they don't, the stain gets stuck. So gram-positives have a thick cell wall. And this is important because a lot of the drugs that we use to treat bacterial infections are targeting this cell wall synthesis or breakdown. And so it's important to know whether something is gram-positive or gram-negative. So this one's a gram-positive. And so Staph aureus is actually part of the normal flora of the human body. So why is it a pathogen? Well, it has to do with when it's in the wrong place at the wrong time. So if it's normally on the skin and there's no cut, it's fine, but it can cause skin infections. It can also be present in your nasal passages, so it can cause sinusitis. It can become, if it gets in the body, it can cause sepsis, which is an infection in the blood. It can also cause osteomyelitis. And so that is an infection associated with bone. And that's where something that's called a biofilm is important. And so just like post-surgical infections, especially associated with implants, bacteria can really firmly adhere to either the bone or the implant. And basically, they form a cement. The bacteria secretes some factors and they form a cement and they sit in there. And this makes them extremely resistant to antibacterials, so antibiotics that we normally treat. Even if the bacterium was susceptible to that antibiotic, the antibiotic can't get to the bug. So once you have a biofilm, it's really difficult to treat. So that's why this approach that they're pitching in this paper to try and combat associated infections with implants is so important. So this biofilm is one of these things that we call a virulence mechanism. But virulence mechanisms in general are just ways to overcome immune responses. And what's cool, and I'm sure you know a lot about this from the virology aspect, when we study how pathogens overcome host defenses, we learn a lot about the immune response itself. Mm-hmm. And so in this case, we know that this particular bacterium, Staph aureus, can produce toxins. And these are fodder for future episodes of immune, but the toxin that they produce is called protein A. And those of you who work in a research lab and have ever done immunoprecipitations, you might know this protein because it binds to antibodies really, really well. And we use it to pull down antibodies and anything that they're associated with in the lab. But why this is so important is that it binds to what's called the FC region, or the crystallizable region, or the constant region of antibodies. And we've talked about antibody structures before in previous episodes. But it binds to this FC part, and that basically turns the antibody the wrong way so it doesn't bind to the bacterial surface and then can't do things like activate complement. So basically the bacteria just grabs onto the antibody the way a cell normally would and prevents it from being able to inactivate the antibody. And if you want to learn about complement, we did talk about that in episodes Immune 7. And so that this idea that these bacteria can produce all these different virulence mechanisms is really cool. And another virulence mechanism that Staph aureus can use is called a super antigen. And I thought that's a really cool thing too. And this super antigen basically just activates every T cell in the body, non-specifically, which might sound at first like a really good idea, but what it does is cause systemic toxicity because you activate all the T cells at once and they start secreting cytokines. You get a cytokine storm. And so between protein A superantigens and then these biofilms, this S. aureus has lots of different mechanisms to overcome immune responses. It's also important to know that there's no vaccine for S. aureus. So if you want to try and treat people with this disease, we have to rely on currently antibacterials or antibiotics. And there's a problem there is that some bacteria develop resistance. And so this is another virulence mechanism where a bacteria can produce proteins that inactivate the antibiotics that we use. So one that we use is penicillin. So you probably know penicillin pretty well. And bacteria can produce a penicillinase and that will cleave penicillin and inactivate it. And one of the things that bacteria do is they like to trade these things on the black market, right? So they have these plasmids that have these biotic resistance genes, and they trade them around like trading cards. And so bacteria can develop resistance to new antibiotics that are out there, or they can pick one up from another bacterium. And so they pass these things around. And this is why physicians tell you have to complete your course of antibiotics, because if you stop early, some of the bacteria can escape trade resistance genes and develop resistance. And so this is a big problem. We now have a whole arsenal of antibiotics in which are becoming obsolete because the bacteria that we're trying to treat are no longer susceptible to those antibiotics. And
it's remarkable that we're alive, right?
It is. But, we just have to develop new ones if we want to keep surviving this. What are the major medical advances that have lengthened the lifespan of the human population? One, vaccines, right? Mm-hmm, so that's the major way to protect against viruses, antibiotics, and hand washing and sanitary practices.
Yeah, public health measures, right? Yep.
Yeah, those three things are three defense mechanisms against major outbreaks of microbial infection. So Staph aureus in of itself is not so much of a problem, but when staph picks up all these different genes that they've traded with other bacteria, they become multi-drug resistant. And one of the ones that has a special name is called MRSA, or methicillin-resistant Staphylococcus aureus. And this is a major problem, especially in hospitals. So in hospitals, there's a lot of this infection that gets passed around. So, people, when you're sick, if you don't have to go to the hospital, don't, because you might get an infection while you're there. These infections are called nosocomial infections. And in the case of MRSA, it's probably not just one gene, but it's a suite of genes called the cassette, and they trade this back and forth, and they become resistant to multiple antibiotics at once. And so with MRSA, there's basically one last resort antibiotic that we can still use against this microbe, and that's called vancomycin. And the CDC has clamped down really tightly on any distribution of vancomycin except for this MRSA infection, because if MRSA becomes vancomycin resistant and gets around, we literally have nothing to fight this bacterial infection, and that's going to be really bad. And we do know that there are a few vancomycin resistant Staph aureus around. But so the bottom line here is that this MRSA or MRSA infection is difficult to treat. It can also predispose people to other infections. And so this idea that we need new creative ways to treat bacterial infections that don't rely on the repertoire of antibiotics we have because the bacteria are becoming resistant is really important. And NIH has highlighted this as a high priority area. And if you're interested, we'll put a website in the show notes that has up-to-date information and new research that's being done in this area of antimicrobial resistance that's so very important. And so this antimicrobial resistance and infections and MRSA is really a problem, especially when we're talking about post-surgical implant infections. And this is because, as I mentioned earlier, you only need a few of the bacteria. If you're putting an implant in, if a few of them get associated with the implant, they make this cement, they colonize. And one, it's really hard to detect because the bacteria are not free floating in the blood. They're not free floating in the tissue, so it's hard to detect it. And if it is detected, it's extremely difficult to treat. And so with the aging population and many, many more people getting implants, this is really a major problem. And so if we talk about what does our immune system do to detect these microbial infections, we use primarily innate immunity. And this is through pattern recognition receptors. And we talked in 2 episodes ago about the VLRs in lamprey, and TLRs are PRRs in mammals. So there's a lot of acronyms here. But pattern recognition receptors, or PRRs, are many, many different classes of receptors that can detect conserved structures within microbes. Do you know any structures that they recognize?
Well, they do recognize RNA, like double-stranded RNA, or
that's right.
RNAs without caps. They can recognize in bacteria things like lipopolysaccharide, right?
Yep.
Can recognize proteins. They can recognize DNA.
Yep. So this class of receptors that are all different kinds of receptors can detect nucleic acids, lipids, proteins, so all the major building blocks of life, and they can detect them from bacteria, viruses, parasites, fungi, you name it. And there aren't a lot of these. They're conserved, but they have a broad binding specificity. And so the idea here is you encode a few of these in your genome, and you can basically, I describe it as detecting whether it's an enemy or a friend. And there's a frenemies, I guess, too. We don't have to go into that in too much detail. But these receptors are tasked with saying, is there danger or is there an infection? And if there is, they initiate the inflammatory response and host defense. So these receptors are primarily expressed on the cells that are required to defend the body upon initial infection. And these include macrophages and dendritic cells, and we've talked about those. But they're not exclusive to those because, as you mentioned, RNA can be detected and viruses, and viruses can get into any type of cell. Each virus has its own selective cells that it goes into, but pretty much any cell could be infected with a virus. So every cell has to be poised with these pattern recognition receptors. And so that's an interesting thing that has changed in the field over the last, 5-10 years, as we thought these were exclusively expressed on specific innate immune cells. But we now know that there's the need and capacity to detect infection in pretty much any cell in the body. But when we're talking about this MRSA or staph infections, they're primarily recognized by one of these pattern recognition receptors called toll-like receptor 2. And toll-like receptors are TLRs. And there are many, many different TLRs. There's a somewhat different repertoire expressed in mouse and human, which complicates some model disease in mice. But generally, TLR2 is relatively conserved, and TLR2 binds and dimerizes with another TLR. It can be TLR1 or TLR6, and they form a competence signaling complex. And so all of these TLRs require what's called dimerization, and TLR2 is the only one that heterodimerizes. That means it binds to a different TLR. The rest homodimerize. That means it binds to itself. But this dimerization, or two components of the receptors coming together, is required to initiate signaling. And so, as you mentioned, some of the different things that can be recognized by bacteria, or of components of bacteria that are recognized by these TLRs, include peptidoglycan, which, if we go back to what I was talking about before, is a component of the cell wall. So when you have a gram-positive and it has a thick cell wall, it has a lot of peptidoglycan. If that peptidoglycan is released, the TLR2 dimerized with TLR1 or 6 can initiate inflammatory response.
So when you say an inflammatory response, how would that be antibacterial?
Right. So there is a production of a lot of inflammatory cytokines that activate cells like macrophages that can eat the bacteria. There's production and activation of complement to coat the bacteria for killing. There's also production of antimicrobial peptides that can bind and lyse the bacteria. There's probably other mechanisms that I'm forgetting about. Activation of the adaptive immune response to produce antibodies that could bind to the bacteria. And if the bacteria reside inside cells, you could induce specific T cells that can then recognize the infected cells and lyse them as well. So there's a lot of different ways that could be activated.
And hopefully in the future we'll get into some of those as well, right?
Right. And one of the things that the inflammation does is prime the site of infection for access by other immune cells to that site. So the inflammation causes vasodilation, so the blood vessels get leaky and the immune cells can get out of the blood easily into the tissue. And this is called chemotaxis or leukocyte infiltration. And one thing that Staph aureus does is prevents that. So if the staph infection is allowed to exist for very long, it's going to shut down the ability of those immune cells to access that site. So the idea of rapid detection and killing these bacteria before they cripple the immune cell access is really important, because once the bacterial infection, if it becomes systemic or septic, it can be rapidly fatal. And the antibiotics aren't going to help very much at that point. And so coming back full circle, these bacteria, these MRSA, have developed this multi-drug resistance. So you try to treat with antibiotics, you can't cure the people of this infection. So this is a major problem. But what this paper that I'm going to summarize for you briefly is doing is they took what we know about these pattern recognition receptors, and specifically toll-like receptors, and they engineered cells to use these TLRs to detect the infection and then produce a molecule that's independent of antibiotics to directly kill and lyse the bacteria. And so they call this, instead of a detect, deflect, destroy policy that the immune response typically uses, they propose to use a direct sense and destroy strategy. So eliminating all of the other inflammatory components that we talked about and ways to activate the immune response, just activate and kill. And so what they did was engineer a synthetic genetic circuit that expresses a protein called lysostaphin under the control of TLR signaling. So lysostaphin is something called an endopeptidase, and that cleaves the peptidoglycan that holds the bacteria together. So it works a lot similar. It's similar to a way an antibiotic would work, but it's enzymatically active and can cleave and kill the bacteria. And because it has this enzymatic activity, it's actually more active against the bacteria than the best available antibiotics. So it's a really great idea. The problem is if you inject lysostaphin in the body, it's rapidly cleared and it doesn't get to the right site of infection, and it doesn't work very well. So expressing this in specific cells would work great. And if you link it to TLR signaling, as soon as the TLR detects the infection, now you have this closed loop that will cause those detecting cells to secrete the antimicrobial right where it needs to be at the right time.
Do you think that one could, or a bacterium could develop resistance to lysostaphin? I'm sure people have looked at that, right?
I don't know. I didn't look that up. I suppose it's possible, but you'd have to change the molecular structure of the peptidoglycan.
Right.
And so I'm not sure how easy it is for a bacterium to do that.
Here's a paper from 2007. Lysostaphin-resistant variants of Staph aureus demonstrate reduced fitness. So maybe they wouldn't make it, right?
Right. So this is actually a really interesting point, because for those aficionados who listen to this, there's a man named Charlie Janeway, and he was at Yale, and he proposed this initial idea of innate immunity many years ago. They called it the dirty little secret of immunologists, that you have to put something into a vaccine to make it work. And that stuff was components of microbes. And we now know that this is, you need this innate activation to have any kind of immune response. And so when he proposed this, he was very, very clever and really amazing in producing this theory. But he basically said that if we were to have this innate immunity, it must target things that are part of microbes that are absolutely required for life. Because if they could swap it out like viruses do to evade antibody response, then they would have developed the ability to evade this. So, and since we are going to have encoding this innate immunity in not the same way as adaptive immunity, so it's limited in its ability to recognize things. If that's the case, then it must recognize things that are one, conserved, and two, crucial for life. And a lot of that has panned out to be true.
But maybe he didn't anticipate the antagonism, the extent of antagonism that we see, as you mentioned, in bacteria, and certainly in viruses. Almost every one of these sensors and the signaling pathways can be targeted by various proteins. So there's a way to get around it.
There's always a way. I think why they haven't completely overcome the innate immune system is that there is a redundancy in the system. And so if the virus evades one mechanism like RIG-I, there's backup mechanisms to detect a viral infection.
Yeah, there's also countermeasures, right?
There are. Mm-hmm.
It's an arms race.
It's exactly an arms race. I was just going to say that. It's whatever the virus comes up with, the host comes up with something else to combat it. And it's a continuous war back and forth.
And as I said before, it's really remarkable that we're around because, we have these great defenses, but viruses, bacteria, fungi, they can evade it in so many different ways, yet it goes back and forth. So there are ways to survive. Obviously, because viruses and bacteria couldn't wipe out all their hosts, right? Otherwise, they wouldn't be around. So there has to be some kind of moderation, some in-between point.
Yeah, we anthropomorphize viruses and bacteria, but the general idea is, if they wiped out too fast, they wouldn't have a host to live in. So they want to tickle the system and make it through to some point, but not overdo it. So there's a balancing act. I guess, it's selective pressure, right? If a virus develops that wipes out the host before it can transmit, it's not gonna last very long. So you select for the ones that are the middle ground, and then they just go for tit for tat back and forth to survive. So generally summarizing this paper, they made this thing they call the InfectSen network, which is basically just an infection sensor. And they did a lot of work. I really liked this paper. They were very thorough. And there's a lot of optimization and selection. But the bottom line here is that they get constitutive expression of TLR2, TLR1, TLR6, and a molecule I haven't talked about, which is CD14. And there's, all this nomenclature, but suffice it to say that CD14 is a co-receptor that binds to some things and amplifies the signal. And they use that as the sensing cassette. So they took what we know about the ability of these receptors to detect staphylococcal infection, and they put them in these cells, and then they link them up with transcription factors that are important for driving inflammatory responses. And so those would be the ones that are activated through these receptors. And those are primarily proteins called AP1 and NF-kappa B. And so these cells have this transcription factor cassette. So it will be activated when the TLRs are tickled on the surface. And then they took this and they put it upstream of the protein lysostaphin. So what they get is if there's staph infection, the staph is going to activate TLR2. The TLR2 is going to activate the transcription factors, and the transcription factors are going to drive lysostaphin. Lysostaphin is going to be secreted and it's going to kill the bacteria. So it's this idea of this closed looped thing going on. So they had this ability to express these proteins in the cell constitutively, the transcription factor cassette, and they can derive this lysostaphin. But if it only worked in one model cell, it wouldn't be very useful. So they tried it in a whole bunch of different cell types, and they settled on the innate immunologist's favorite little cell, which is called the HEK293, or the human embryonic kidney 293 cell. And they just showed beautiful characterization that in this particular cell line, when they express this whole cassette, they could get activation with live or heat killed, or even little components of the peptidoglycan of Staph aureus. They showed dose responses, time courses, and 100 bacteria were enough to trigger these cells in vitro. And really importantly, there was low leakiness, because if you had constitutive production of lysostaphin at a low level, that's a recipe for developing resistance if it were going to happen, right? And because of the way they engineered the system, it was a really rapid response. So if you put the bacteria in this tissue culture, within 4 hours, you could detect the response out with a fluorescent reporter assay. And it was dose dependent. The more bacteria you put in, the faster the system responded, which is what you would want to happen, because if you have a high level infection, you need a lot of lysostaphin really early to control the infection. If you have a low level infection, you could produce lower levels of lysostaphin and prevent the infection. So it's a really neat system
and
there.
The cells seem to be fine, right? Which
the cells are fine.
Yeah.
Yep. This is not it's the lysostaphin is not going to do anything to a mammalian cell. They don't have peptidoglycan.
Right.
So it's targeting the bacterial cell specifically.
And they are secreted. The lysostaphins are secreted, right?
It's secreted, yeah. Because they could, later on, they put it in vivo and they could detect, well, they didn't do that in the blood, but they could detect the lysostaphin secreted by the cells in the culture media. And so they showed a really interesting figure that I thought was really cool. And that was, if they add the bacteria in, they turn this reporter on. They could put a reporter in place of the lysostaphin and then just watch this over time in real time. And if they put the bacteria in, the reporter goes on. If they wash the bacteria out, the reporter goes off. They put the bacteria in, the reporter goes on. They wash the bacteria, the reporter goes off. So it's really tightly controlled, which is also a characteristic you would want if you're going to put this, like, for example, embed it within something or attach it to an implantable device. You want it to be on when you need it and not on all the time. Because the reality is the immune system will probably eventually develop a response against the foreign protein.
Of course.
So you want to control where and when it's made. So at that point, they made this modified lysostaphin that was really stable, and they confirmed that their sensor cells would produce this and secrete it, and that it was active against the bacteria in the culture. And they show that they could kill the bacteria in vitro over 6 logs. So they could add very high levels of bacteria into the tissue culture dish, and those cells would be fully protected through this system. And of note, they had to use a different staph, because I would assume they probably didn't get permission to use methicillin-resistant Staphylococcus aureus. So they use a different staph called Staph carnosus to do all their testing. But they say that that's very equivalent to the MRSA, so it would be relevant. And the one cool thing that they did do was so they have their cells culturing, and they add the staph, and the lysostaphin gets turned on and kills the staph, but then they leave it for 3 days, and there's no bacterial growth. That's complete elimination of the infection. Because even if you have a few bacteria sneaking through, your culture would be overwhelmed in a few days. So this is a really, really powerful and robust system to control the bacterial infection.
Yeah, people have explored this kind of strategy using phage lysins, right? Similar idea, but not with this approach. As you said earlier, they try to put the protein in, and that's immunogenic, and it doesn't last very long, and so forth. But this is lovely because these cells continue to produce the protein.
They do. Yep. In the local region of where you're interested, because they're going to get to that in vivo, right? So you might be asking at this point, that's great. Everything's in vitro. You can kill a lot of bacteria, but what the heck happens if you actually go into an animal? Is this just all well and good, but irrelevant? And so if you just put the cells into the body, they're probably not going to last very long. These HEK293 cells,
cleared, right?
Yeah. Do you know why?
Why they're cleared?
Yeah. Why would they be cleared really fast?
Well, they would
if you put them in a mouse.
Well, we have this reticuloendothelial system that would, if you put viruses in, they're cleared by it. So cells would certainly be cleared, right?
Yep. So the cells could just be flushed out, but they're also going to be recognized immunogenically because they're human cells. You put human cells into a mouse.
That'll take a while, right?
And they'll reject them.
How long would that take?
I think they reject them pretty quickly.
How long would you say? I mean, 10?
So, if you take one mouse skin and you transplant it onto another mouse skin, within 14 days, they reject it.
So, yeah, that's 2 weeks. It's about right for an adaptive response, right?
Yeah.
So, but it's not immediate. But there
can be immediate rejection. So it has to do with major histocompatibility and minor histocompatibility antigens. So if you mix two people's blood together, they attack each other right away. So you don't need the development of the adaptive immune system. So that's why you do testing to do transplants, right? Because if you put someone's spleen into me and I'm not matched, I'll actually reject it as soon as you connect the blood. So it can be very rapid. So if you put these human cells into this mouse, it wouldn't work very well, but they got clever and they encapsulated them into these little microparticles. And so the cells are protected from the immune system, but the cells can make and secrete things that will then be released out of the encapsulated alginate.
Okay, so it's
cool.
an algae-based capsule, so it's permeable to the lysostaphin, right?
Yep. So, yeah. So this is the engineering approach. And so they are able to inject these encapsulated cells into mice, and they did it intraperitoneally just for the sake of ease to do this type of study. They injected the encapsulated cells, and then they injected the Staph carnosus into the mice. And what they did was the cells they used this time had a reporter instead of the lysostaphin because they just wanted to see if it would work. And they looked in the blood to see if they could see the reporter. And they could. And it was dependent on TLR2. So if they left TLR2 out of their cell construct, there was no response. So you needed the TLR2 to detect the Staph carnosus, which then induced the transcription factors, which produced the reporter. The reporter was secreted out of the encapsulation and got into the bloodstream, and they could bleed the mice and detect it in the blood.
How did they put the Staph into the mice?
They injected IP also. So they put it in the right place. They put it in the same place as they put the cells to make it easy. So that's all well and good, but that's all in like a soup in the belly of the mouse. But what would happen if you did an implant? So they have this cool thing that's this tissue cage, and I looked it up a little bit, and there's a cool picture associated with the paper if you're interested in looking that up. Maybe we could use that as the picture for this particular episode. But it's a Teflon tissue cage, and it's got a whole bunch of different holes in it, and it lets fluid in and out, and I think it might let some cells in and out if they crawl, because phagocytes will, move in and out of relatively small spots. But what they can do is they can implant this into the mouse, and then they can use a needle and they can harvest fluid from inside the cage, and so they can tell what's going on there, and they can see if there is bacteria swimming around in the cage. They can tell if the sensor's been triggered, and then they can measure the level of infection. And in this case, they used the mice that were expressing constitutively the lysostaphin as a positive control, and then they also induced produced encapsulated cells and put them in with this tissue cage and see what happens. And the other thing with this tissue cage that allows you to do that you can't do with just injecting into the peritoneal cavity is you can look at biofilm. So if the bacteria become associated with this cage, they'll form the biofilm, and so you can harvest the cage and you can look at how much biofilm associated bacteria there are. So they were able to see a couple of things. They could see the lysostaphin go up, so they could measure that. They could measure the level of infection, and they couldn't detect it if they had this system in place. So there were no free swimming bacteria. And when they looked at the bacteria associated with the cage, they were also eliminated. And it all depended on the TLR2, and it depended on the production of the lysostaphin. So the system seems to work. And then, as you said, if this really mattered, they would do this with the MRSA, and they did. So they were able to infect some mice with methicillin-resistant Staph aureus, and the encapsulated cells protected the mice. And in fact, the encapsulated cells performed better than treating the mice with vancomycin or just with recombinant lysostaphin injected into the body. And they didn't detect any biofilms, and there were what's called treatment failures. If they treat the infected mice that have with like vancomycin, there are a certain percentage of them that they're going to fail and they'll still get infection. But if they use the sensor cells and that produce the lysostaphin, they didn't have any failures. But if they treated the mice with vancomycin or treated them with recombinant lysostaphin, there were failures. So this seems to work better than the best antibiotic. I'm sure there are caveats to the whole thing, but generally what they did was they used genetic engineering to make cells that could sense really low bacterial loads, and it worked both in vitro and in vivo. And they engineered these to produce this highly specific and really active antibacterial protein that prevented the bacterial infections and prevented biofilm formation on implants. So the idea is that this would be difficult or impossible, as you found the paper that showed that the resistance was not compatible with bacterial survival. So if you can detect these, it seems like it would be a really good way to work, and it supports the idea that there's lack of evolutionary evasion of pattern recognition receptor detection.
There are, of course, other obstacles. And they point these out. One of them, I think, you can't use HEK293 cells, obviously, right? So you're probably going to have to take, like as in CAR T cell therapy, you're probably going to have to take cells out of the patient and put these constructs in them and then infuse them back. And, it depends. I guess if you're putting in an implant, you could put them with the implant or something like that, right? How much it's going to cost is another issue. Of course, the CAR T cell therapy is huge.
No idea.
And this is going to be used far more than CAR T cell therapy, right? Because there are a lot more infections associated with implanted objects and so forth. So that's another thing. And as they say, you're going to have to monitor the cells and know how long they last, if you can take them out or shut them off in some way or give new ones. And,
yeah, I suppose you'd have to engineer a kill switch, right? Yeah. Just like they do for CAR T cells. And I guess the one thing that you don't have that you have to do in CAR T cell therapy is you don't have to eliminate receptors that are already present. Because in the CAR T cells, we want them to not have the endogenous receptor so that they're really using the CAR T receptor so that they're not generally activated. And I guess they do that to some extent. But here it wouldn't matter. You already have cells that express pattern recognition receptors. You could just put in the sensor reporter or the reporter, the transcription factor cassette, and the antimicrobial.
The other concern is that you're going to make antibodies against lysostaphin, right? So what's going to happen? The first time it'll be okay, but then it may not work. So maybe they have to make antigenic variants, right? That could be used to. That's being done with viral vectors. You can engineer the capsids so they're different enough so they're not cleared. So maybe you would have to do something like that, or maybe it's a one-shot deal. I don't know, right?
Could be a one-shot deal. I guess the one benefit would be is if this is closely associated with an implant, even if you generated antibodies, if these cells are producing this lysostaphin, there's probably enough of it not bound by antibodies to be able to bind to the bacteria and kill them. I don't know. I guess you'd have to just test it. Maybe one way one could do that is pre-immunize a mouse with recombinant lysostaphin first, and then see whether your encapsulated cells actually work, where there's antibodies on board.
No,
but they didn't do that.
Plenty to do, and this, as you say, looks great so far, and so therefore it should go forward.
I just love these really creative ways of thinking outside the box to take what we know about immunology and make something totally different. I love this kind of approach. I think it's really
immunology and of course molecular biology, knowing how to put control plasmids together with sequences that are regulatory and all that stuff. I mean, this is just decades of fundamental work that then all comes together. It's just great, right?
Yeah, that's the key, right? Fundamental work. If we didn't have all of that groundwork laid, you wouldn't be able to come up with these great ideas that are therapeutically.
The key is that often you don't know it ahead of time, right? Just like CRISPR, we had no idea what it would be good for, yet people just said, hey, let's figure this out. That's basic fundamental research, and it turned out to be really useful, but you're never going to get that if you don't look, right?
All we can do is keep standing on the soapbox screaming and make all of our colleagues remember that.
I tell people you have to have examples in your head all the time to tell people in an elevator. If it weren't for this, we wouldn't have this, right? And you make it.
Yes.
Serendipity
because when they get
so
in that
important.
room and do those,
sometimes they're focused translational stuff, which you say we're going to go after this, fine. But for the most part, all the stuff we talk about is accidental, right? CAR T-cell therapy is based on a lot of accidental research done for years, right? So you have to keep doing that. And
it's discovery.
Non-scientists don't quite understand. Of course, listeners of this show who are not scientists, they do understand because they've sought out these podcasts. But a lot of people don't understand this idea that you should allow people to investigate what they think is interesting. And out of that comes stuff that can help human health.
Yeah. There's a lot of examples where people talk about the cell phone and other things, technologies that people use every day. It's better to use those examples because people, if you talk about CRISPR, they don't have enough knowledge to be able to understand the implication of that, right? But if you tell them you wouldn't have your cell phone if they weren't doing basic research,
I would say,
then they kind of get that. So we also have to tailor what, our science communication message to something that people can sink their teeth in and understand.
Cell phone's a great example, but then I would take it a step further. So you wouldn't be alive to use your cell phone if it weren't for, vaccines and antibiotics and antivirals and public health measures. So people need to understand it.
Yes.
CRISPR isn't there yet, but I think one day it's going to be awesome. But we can point to so many other things. Recombinant DNA has made available drugs that we didn't have before, and these save lives. So, the cell phone is great. I think it's a cool example of science and technology. But if we were dead, there'd be no cell phone. So, I think everything stems from life science research. Of course, I may be biased. You and I are biased,
I
right?
Agree. I think we're biased.
Anyway, that's a great paper. Thank you for doing that. I'm sorry Steph missed it, but
it's fun.
Maybe she's listening and save your questions
She'll
for
next
time. Steph, we speaking of questions, we have some cool emails from our cool listeners. Why don't you take that first one from Niraj?
Sure. Niraj writes, "Hi, immunomodulators." I love it. I should get it's too long for a.
Do you have a science license plate? Why don't you get immune?
I don't have a vanity plate. I don't know. I should.
I doubt
Nobody
it, but
has
listen,
it yet, do they?
I interviewed a guy last week who works on CRISPR. He just moved to Colombia, and I said, "Why don't you get a CRISPR license plate?" The next day, he got it. New York State, he's got CRISPR.
Oh.
Isn't that great? So,
that's awesome.
Why don't you can go online and see.
That's awesome.
Cindy, you can go online and look for immune because that would be cool.
That would be really cool. Then I'd have to take a picture. Then we'd have to be somewhere. So
that's easy. We're only like 500 miles away or 400 or something. Yeah, that's no problem.
All right, back to this, immunomodulators. It's been a while since I wrote in, which is amazing because we've been on a little less than a year and this person has written in more than once, so that's great. It's been a while since I wrote in, but thanks for the wonderful discussion on the uniquely structured lamprey and hagfish antibodies. In relation to this, I had thoroughly enjoyed the TWIV 467. Steph already put it as part of her pick of the week. And it has intrigued me towards the notion that maybe we know where the TLRs evidently came from and why some of them have such high specificity for microbial ligands, TLR4 for LPS, etc. But then listening to the discussion on the current immune episode completely blew my mind away. To be separated by millions of years and to still recognize the same antigenic epitopes on a viral protein with high specificity is bizarrely amazing. This is why being in science is so rewarding and gratifying. At times, one gets to make factual discoveries that totally make you realize that even after years of toiling, you know so little. Smiley face. What is that?
Yeah,
is that a shy smile? Embarrassed smile? Personally, for me, that's the single biggest driver to me to be in research. A chance to work on exciting projects and discovering the unknown. Thus, I just wanted to thank you for your efforts in informing the broader audience, and especially Professor Racaniello. Whenever you feel disgruntled about having to manage the organizing of these podcasts, please keep in mind that the listeners like me are better informed due to your tireless efforts. I have learned a huge deal by listening to the Twix series of podcasts and now Immune. So please continue the great work. And when motivation runs low and frustration is the only emotion, remember the first lines from Robert Frost. The woods are lovely, dark, and deep, but I have promises to keep and miles to go before I sleep. Although I do hope you don't discount your sleep too much for the sake of podcasting. I do hope that you and your esteemed team members will continue to enrich the common man with pearls of wisdom that are discussed on these podcasts for many years to come. Best wishes, Niraj. I second that. I know you get frustrated in the organization of these things, but really, it's amazing. I can tell you, I was listening to the TWIVs where you were traveling for about a month and a half, but each one was so interesting with all of your guests that you had on and the way it was organized. It's so worth all the efforts that you put into this, and I am so privileged to be part of this with you.
Well, thank you. Thank you, Cindy. Thank you, Niraj. I will, of course, continue. There's no question, but I do like to express my frustrations from time to time. But
if you don't, it'll boil up and you'll pop.
No, I love doing this, and I will continue to do it long after I don't do lab work anymore. And that's something I'm struggling with. You know, can I be an effective science podcaster without having a lab? And I'm sure I can, but as you might guess, it's kind of a weird thing, right? As you spend your life doing.
We feel like, we hang our credibility on a currently successful, thriving lab, and it's hard to pull away from that. It's hard to say, could I give that up and be more worthwhile in a different arena?
Right.
We don't know because we don't know anything else. It's all we've ever done.
You won't find out until you try, right? So even though it's scary, and for me, I'm probably 4 or 5 years. That's it for my lab, but then I'll continue to podcast, and I just have to deal with it, right? I have to deal with, and we'll see. I'll let you know if people listen after I stop doing science. I have a feeling they will, but
I think they will.
All right, the next one's from Mike. I want to say I agree with the person who suggested you start your podcast with a summary of what you did that day at work. Sounds like a good idea for those of us, speaking for myself, who haven't spent a whole lot of time in a lab, or it has been many years. I find it interesting to hear what people are doing and how they are using the new technologies out there. Thanks for a great show, Mike in Oregon. So there you go, Cindy. If I close my lab, I won't be able to tell Mike what I did. Well, I'll do things that he doesn't care about.
Like what?
Yard
work and reading a book.
I edited this podcast before doing this one, stuff like that, or I packed for going on the next podcast. But I will have other people on every show, of course, that will still be doing science and who can do that. I think this is a good idea. Maybe the next time when we record Immune, we'll tell you what we did. Let me put it in the beginning.
It might be a little weird though, because if it's all of us people who have been running a lab for a while, it may be, well, I reviewed a manuscript and I worked on a grant. I read 3 papers and I talked to a student. But that would be the fun part, right? I talked to a student and had some really exciting data.
Well, Steph here her career is moving through the early stages, so she'll have a different perspective.
Yeah.
But for the most part, most of us on all the different podcasts I do are pretty senior, so we're doing the same things.
Yeah. Okay, so Bob writes, no sooner had I finished listening to Immune number 9 than I ran across notice of newly published research on type 1 diabetes involving BCG treatment. And there's a link to a paper. And this paper is about immunometabolomics, which is a new hot area of immunology. And he says the panel's discussion of the first letter read on the podcast that of Steve Lutkin, asking about the efficacy of BCG vaccination, was immensely helpful in understanding this paper. I'm very interested in the subject of type 1 diabetes as my eldest daughter suffers from this. Sorry to hear that. Thanks for the informative podcast, Bob. So we hear a lot about diabetes, and it affects a lot of older individuals, especially people with who are obese or overweight. And that's type 2 diabetes. And type 1 diabetes is the insulin-dependent diabetes that's actually an autoimmune disease. So we could cover that on a future episode because we haven't really talked about autoimmune diseases yet, and they're really fascinating. But this particular paper is interesting, and I won't go into great detail of it right now, but it's this idea that there's a shift in the way glucose is used and metabolized in the body. And so we know now that this metabolomics is really, really important for regulating immune responses, and that it is regulated by immune responses and can dictate the type of immune cell function there is. And so there can be a shift from oxidative phosphorylation, for those of you who remember your biochemistry, to glycolysis. And the paper shows that this changes the way glucose is metabolized and lowers blood sugar. And so we know from a number of different people's work, including Erica Pierce, that this metabolic shift can accompany changes in the functional profile of cells. And for me, that it's interesting because it does this for macrophages. So if you TLR stimulate macrophages, it drives what we call an M1 or a classically activated macrophage. And those ramp up this Warburg effect and have high levels of glycolysis. And so you can have different ways in which the glucose is used. And that seems to have something to do with the development or treatment of type 1 diabetes, this autoimmune disease, which is really cool. So thank you for sharing that.
Alberto writes, hello, twimmunocytes. That's a good one.
Yeah.
My name is Alberto. I'm a postdoc studying the human immune system in type 1 diabetes at the University of Pennsylvania. In my graduate work, I used mouse models to understand how T cells fight tumors. I became inspired by your discussion of lamprey immunology to write and hear your opinion about recent papers that compare specific pathogen-free mice with feral mice caught in the wild or mice bred in pet shops. Here are links to some of the papers, including one where I am co-author. It sends 3 papers. 1, wild immunology assessed by multidimensional mass cytometry. The other one, normalizing the environment, recapitulates adult human immune traits in laboratory mice. And finally, we have the comparative immunology of wild and laboratory mice. He goes on to make a long story short, the immune system of mice with a diverse history of microbial interactions is very different from the immune system of specific pathogen-free mice, which are usually shielded from these interactions in the clean laboratory environment. The frequency, phenotype, and function of almost every type of leukocyte, CD4 plus and CD8 plus T cells, NK, B cells, macrophages, is different in dirty mice compared to clean mice. This difference is not due to divergent genetic backgrounds because it can be recapitulated in black 6 mice if they're exposed to dirty mice in co-housing experiments. Having a more mature immune system can make these mice good models of how the adult immune system operates when faced with challenges such as infections, immunizations, autoimmunity, and immunotherapies. However, there are immense practical barriers to implementing dirty mice as a widespread model for immunological research. I'd be happy to hear your thoughts on the subject. I'm a longtime listener of TWIV and TWIP and was very happy when Immune started. You're doing a fantastic job. So, Cindy, you have some thoughts on this?
Well, I think that, everything Alberto said is right and is a little scary because we have 50 to 100 years of immunology in which we've studied progressively cleaner and cleaner mice. And then we have these disparities with what we find in mice is not always translational to other animals or to humans. And so what's the why? What's going on? And you're right. If we make these mice dirty, give them co-infections or give them infections that we would, or take the mice from the wild where they do already have these onboard infections, including a number of parasites, which we often kind of ignore, it changes the immune response when we challenge with viral infections, bacterial infections, vaccines, you name it. So everything we knew about the immune response is kind of in question now until we determine what in fact is different and how that impacts what we already know. Because for years and years and years, we've gotten cleaner and cleaner and better at rederiving microbes, and to the point where we have these pathogen-free mice or notobiotic mice. They have no bacteria. They live in isolator cages. They don't have any skin bacteria. They don't have any gut bacteria. And they don't have a lot of the components of the immune system. They're immature until you colonize them. Then now they respond differently. So looking at the spectrum from notobiotic all the way to catching mice in the wild and looking at them is really important.
Yeah, and it's clear that it makes a difference.
It does.
The studies that have been done so far.
Yep.
And I have to say that it's not just microbiome, but the genome.
Yep.
We have these lines of mice that are completely interbred, right?
Yep.
And it doesn't really tell you the right answers because you could infect one with a virus and get a totally different result from another mouse line, right?
And it gets worse than that because you can have black 6 and black 6. And
that's right. Many different flavors of black 6.
Right. If you've derived them for years and years and years in two different facilities, the epigenetics and slight different polymorphisms and such can change how one lab does an experiment and another lab does an experiment, the type of result that they get.
So we've just begun to work with the collaborative cross mice, which is an attempt to back up and take all these different mice, cross them, and mix up all those genes and then do your experiments. And it's really, really interesting. You can find very interesting mutations that control susceptibility to viruses, responses to infections and so forth that you wouldn't have done if you just stayed with your C57 black 6. So it's really, and that's just the genetic background. Imagine throwing in the microbiome, as we say here. Boy,
right.
Difficult.
It makes it much more complex, but much more interesting and exciting.
For
and
sure.
Also more difficult to tease out what parameter is responsible for the phenotype you see. Some would argue it doesn't matter. If they're, if they're wild, then that's what you should be measuring. so don't break it down and don't try and make them clean. But yeah.
Okay, let's save some email for next time for Steph.
Great.
And how about you and I do some picks of the week?
Sure.
What do you have, Cindy?
So I am linking to a study with vaccines and autism because I love this topic. This is a huge study that was just very recently published. And it once again shows no link between vaccines and autism, but this one took a different approach. So this study evaluated 80,000 mom-child pairs for the frequency of autism in the children that were born to moms that were either received or didn't receive the Tdap vaccine during pregnancy. This is recommended because it will confer some protection to the mom so that she doesn't get the infection, as also allows the mom to make antibodies that can be transferred in the milk once the baby's born. And the bottom line was there were no statistically significant differences in the percentage or number of children that had autism spectrum disease with moms that were either immunized or non-immunized. And when I looked at this study a little more carefully, they're careful not to say this, but I don't know how statistically significant it is, but they actually saw more children with autism in the moms that were not vaccinated versus the moms that were. Now, it was small differences, and they're saying that they conclude that there's no more children with autism in vaccinated moms, because that's really the concern is, does vaccination or immunization cause any disease? And it does not. But in fact, in this case, maybe if the moms get infections, it makes the babies more susceptible. And we know that certain infections in vitro can be dangerous to unborn babies, One of them is Zika, we know, and there's a number of other ones that can transfer transplacentally, or when there's high levels of infection and inflammation in the mom during pregnancy can cause problems in the baby. So
another nail in the coffin.
Get vaccinated.
Right?
How many? I don't think there's any room left in the coffin to nail any more nails in. But yes, and it's so important to get this out there because there are every day, no matter how much we put out the good science, there are mommy blogs and things that just spew this fear mongering to new moms.
It's just
to
remarkable.
Scare them. What's your pick?
Well, my pick is a website called Unpaywall.
Unpaywall.
This is a database of so far 19 million scholarly articles, scientific articles. So the makers of this website have gone out and looked for all these science articles that are open access or accessible to you without having a subscription. So you can search their database for that. And they also tell you about articles behind paywalls that are on legally accessible servers. And so you can go there and search for it. This is a effort of scientists. They started this a number of years ago. It's gotten quite a bit of coverage. It's completely legitimate. It's nonprofit. It's open source. It's endorsed by NIH and Merck and MIT, etc. So if you're looking for open access content, you should try Unpaywall. It's very cool.
This is really neat. So in the time that you were talking, this went up to up 30 articles on there. It's like a ticker there.
Yeah, it's a ticker on
that
the front
shows an increase constantly. The other thing I was going to say is this is really cool because I saw recently on Twitter a discussion by science journalists that were lamenting about the problems of paywall and trying to get access. And so we should put this on Twitter so that they know, they probably know about this, but they might not.
Yeah.
Newer science writers.
No.
They should have access to this.
Because they could probably find the article they're looking for.
Probably.
On this.
That's great. I love it.
Helps. I mean, it's an easy one place to find it. So I like that. All right. That is Immune number 11. It's at Apple Podcasts, microbe.tv/immune. And if you listen to podcasts on your phone or your tablet and a mobile device, you use an app and just search for Immune. You'll find it. Subscribe. It really helps us if you subscribe. That way we can show our numbers growing and you get every episode as we release them every month. And if you want to send us questions and comments, you're totally welcome to. We love answering them. Send them to immune@microbe.tv. And finally, if you'd like to support us, someone wrote into Twiv the other day. She said, I've been listening for years and I decided to support you. I went to Patreon. I noticed there are only 160 people supporting you and you have over 20,000 likes on Facebook alone. She said, I felt so badly. I subscribed right away. So go over to microbe.tv/contribute. We have a Patreon account. We have some other ways you can contribute. Even if you just gave $1 a month, that would really help us. Microbe.tv.
And it's all going to the supporting this podcast.
Yeah, it goes
so that we can bring you the content.
Production costs, travel costs. We don't travel all that much, but when we do, we like to help our hosts travel. And so the more we can get, and ultimately I'm hoping to hire someone to help out technically, so that'll free me up to do more content. Microbe.tv/contribute. Cindy Leifer is at Cornell University. She's on Twitter as Cindy Leifer. Thanks, Cindy.
Thank you. This was fun.
I'm Vincent Racaniello. You can find me at virology.ws, PROFVRR on Twitter. Music is by Steve Neal, stevenealpercussion.com. Thanks for listening to Immune, the podcast that's infectious. Be back next month.